Q1. According to the fluid mosaic model proposed by Singer and Nicholson, 1972, which statement best describes the plasma membrane? A. Proteins form a continuous layer on both sides of a phospholipid bilayer. B. Lipids are embedded within a protein matrix. C. Proteins float within or on the phospholipid bilayer, allowing dynamic movement. D. The membrane consists entirely of phospholipids without proteins. Answer: C. Explanation: the fluid mosaic model states that the plasma membrane is composed of a phospholipid bilayer with proteins embedded or associated with it. Both lipids and many proteins exhibit lateral movement, making the membrane dynamic rather than rigid. Difficulty. Topic: Plasma membrane. Q2. Which of the following membrane proteins is primarily responsible for facilitating passive movement of ions across the plasma membrane?
A. ATPase pump. B. Channel protein. C. Integrin. D. Glycoprotein receptor.
Answer: B. Explanation: channel proteins provide hydrophilic pores that allow specific ions or molecules to move down their concentration gradient without ATP consumption. ATPases are involved in active transport. Difficulty. Topic: membrane transport. Q3. The asymmetrical distribution of phospholipids in the plasma membrane is maintained mainly by A. DNA polymerase, B. flipasase and floppasase, C. hist-1s, D. ribosomes. Answer: B. Explanation: flipasase, floppasase, and scramblasase regulate the movement of phospholipids between the two leaflets of the membrane, preserving membrane asymmetry, which is important for cell signaling and apoptosis. Difficulty. Topic: membrane dynamics. Q4. Which organelle is considered the primary site for sorting, modifying, and packaging proteins destined for secretion? A. rough endoplasmic reticulum. B. lysosome. C. Golgi apparatus. D. peroxisome. Answer: C. Explanation: proteins synthesized in the rough endoplasmic reticulum are transported to the Golgi apparatus, where they undergo glycosylation, sorting, packaging, and targeting to their final destinations. Difficulty. Topic: Golgi apparatus. Q5. Which statement regarding ribosomes is incorrect? A. They are composed of rRNA and proteins. B. They are the site of protein synthesis. C. They possess a surrounding phospholipid membrane. D. They occur freely in the cytoplasm or attached to rough ER. Answer: C. Explanation: ribosomes are non-membrane-bound organelles. They consist of ribosomal RNA and proteins and function in translation. Difficulty. Topic: ribosomes. Q6. The enzyme catalase, responsible for the breakdown of hydrogen peroxide, is predominantly localized in
A. mitochondria. B. Golgi bodies. C. peroxisomes.
D. chloroplasts. Answer: C. Explanation: peroxisomes contain catalase, which converts hydrogen peroxide into water and oxygen, protecting cells from oxidative damage. Difficulty. Topic: peroxisomes. Q7. Which component of the cytoskeleton is directly involved in chromosome movement during mitosis? A. microfilaments. B. intermediate filaments. C. microtubules. D. actin filaments. Answer: C. Explanation: Microtubules form the mitotic spindle that attaches to kinetochores and segregates chromosomes during cell division. Difficulty. Topic: Cytoskeleton. Q8. A mutation affecting the signal peptide of a secretory protein would most likely impair its entry into the A. Golgi apparatus, B. Rough endoplasmic reticulum, C. Lysosome, D. Peroxisome. Answer: B. Explanation. The signal peptide directs the ribosome protein complex to the rough endoplasmic reticulum through the signal recognition particle, SRP. Without this peptide, secretory proteins remain in the cytosol. Difficulty. Topic: Protein targeting. Q9. Which of the following is the most reliable evidence supporting the endosymbiotic origin of mitochondria? A. Presence of cholesterol in mitochondrial membranes. B. Presence of linear DNA. C. Presence of circular DNA and 70s ribosomes. D. Presence of histone proteins. Answer: C. Explanation. Mitochondria possess circular DNA and 70s ribosomes, characteristics shared with bacteria. These features strongly support the endosymbiotic theory proposed by Lynn Margulis. Difficulty. Topic: Mitochondria. Q10. Treatment of eukaryotic cells with colchicine would primarily inhibit Q10. Treatment of eukaryotic cells with colchicine would primarily inhibit A. DNA replication, B. Protein synthesis, C. Microtubule polymerization and spindle formation, D. RNA transcription. Answer: C. Explanation. Colchicine binds to tubulin and prevents microtubule polymerization, disrupting spindle formation. As a result, cells are arrested in metaphase and chromosome segregation cannot occur. Difficulty. Topic: Cell division. Q11. A eukaryotic cell contains numerous membrane-bound organelles. Which organelle is primarily responsible for ATP synthesis through oxidative phosphorylation? A. Golgi apparatus, B. Mitochondrion, C. Peroxisome, D. Lysosome. Answer: B. Explanation. Mitochondria are the powerhouse of eukaryotic cells. ATP synthesis occurs on the inner mitochondrial membrane through the electron transport chain and ATP synthase during oxidative phosphorylation. Difficulty. Topic: Mitochondria. Q12. The primary function of the nuclear pore complex is to A. Synthesize DNA, B. Regulate selective transport between the nucleus and cytoplasm, C. Produce ribosomes, D. Replicate chromosomes. Answer: B. Explanation. The nuclear pore complex, NPC, controls the bidirectional movement of proteins, RNAs, ribosomal subunits, and signaling molecules between the nucleus and cytoplasm. Difficulty. Topic: Nucleus. Q13. Which of the following cytoskeletal elements provides maximum tensile strength to cells? A. Microfilaments, B. Intermediate filaments, C. Microtubules, D. Centrioles. Answer: B. Explanation. Intermediate filaments are highly stable and provide mechanical support, maintaining cell shape and resisting stretching forces. Difficulty. Topic: Cytoskeleton. Q14. Which statement regarding lysosomes is correct? A. They are synthesized directly from mitochondria. B. Their enzymes exhibit maximum activity near neutral pH. C. They contain hydrolytic enzymes active under acidic conditions. D. They lack membrane boundaries. Answer, C. Explanation. Lysosomes contain acid hydrolases that function optimally at an acidic pH, 5, maintained by proton pumps in the lysosomal membrane. Difficulty. Topic: Lysosomes. Q15. The membrane potential across the plasma membrane is primarily generated by A. Diffusion of glucose, B. Active transport of Na and K ions, C. DNA replication, D. Protein synthesis. Answer, B. Explanation. The Na/Kat phosphate actively transports sodium out of and potassium into the cell, creating ionic gradients that establish the membrane potential. Difficulty. Topic: Membrane transport. Q16. Which organelle is mainly responsible for detoxification of drugs in liver cells and is abundant in hepatocytes? A. Rough endoplasmic reticulum, B. Smooth endoplasmic reticulum, C. Lysosome, D. Golgi apparatus. Answer, B. Explanation. The smooth endoplasmic reticulum contains enzymes involved in lipid metabolism and detoxification of drugs and xenobiotics, especially in liver cells. Difficulty. Topic: Endoplasmic reticulum. Q17. During receptor-mediated endocytosis, the protein responsible for forming coated vesicles is A. Tubulin, B. clathrin, C. dynein, D. myosin. Answer, B. Explanation. Clathrin assembles into a polyhedral coat around vesicles, facilitating receptor-mediated endocytosis and selective uptake of extracellular molecules. Difficulty. Topic: Membrane trafficking. Q18. Which of the following statements best distinguishes apoptosis from necrosis? A. Apoptosis always causes inflammation. B. Necrosis is a genetically programmed process. C. Apoptosis is an ADP-dependent, programmed process that generally does not induce inflammation. D. Both apoptosis and necrosis occur through identical molecular pathways. Answer, C. Explanation. Apoptosis is a regulated process involving caspases and ADP, leading to controlled cell death without releasing intracellular contents. Necrosis is uncontrolled and usually triggers inflammation. Difficulty. Topic: Cell death. Q19. Which of the following organelles possesses its own DNA but is not considered part of the endomembrane system? A. Golgi apparatus, B. Endoplasmic reticulum, C. Mitochondrion, D. Lysosome. Answer, C. Explanation. Mitochondria contain circular DNA and replicate independently. They are not derived from the endomembrane system and are believed to have evolved through endosymbiosis. Difficulty. Topic: Cell organelles. Q20. A scientist isolates a mutant cell line in which newly synthesized proteins accumulate in the rough endoplasmic reticulum and fail to reach the Golgi apparatus. Which process is most likely defective? A. DNA replication, B. Vesicular transport mediated by copy-coated vesicles, C. Translation on ribosomes, D. Glycolysis. Answer, B. Explanation. Copy-coated vesicles transport proteins from the rough endoplasmic reticulum to the Golgi apparatus. Defects in copy proteins prevent export from the ER, causing protein accumulation. Difficulty. Topic: Protein sorting and vesicular transport. Freeze fracture electron microscopy is particularly useful for studying A. Ribosome assembly. B. Internal structure of chloroplasts. C. Distribution of membrane proteins within lipid bilayers. D. DNA packaging in chromosomes. Answer, C. Explanation. Freeze fracture electron microscopy splits membranes along the hydrophobic interior of the lipid bilayer, revealing the distribution of integral membrane proteins as intramembranous particles. Difficulty. Topic: Plasma membrane. Q22. Which scientist is credited with the discovery of lysosomes? A. George Palade. B. Christian de Duva. C. Camillo Golgi. D. Albert Claude. Answer, B. Explanation. Christian de Duva discovered lysosomes in 1955 through cell fractionation studies. He was awarded the Nobel Prize in Physiology or Medicine in 1974 along with Albert Claude and George Palade. Difficulty. Topic: Cell organelles. Q23. Which cellular component is primarily responsible for maintaining turgor pressure in mature plant cells?
A. Mitochondrion. B. Central vacuole. C. Golgi apparatus.
D. Peroxisome. Answer, B. Explanation. The large central vacuole stores water and solutes, generating turgor pressure that maintains cell rigidity and supports plant tissues. Difficulty. Topic: Plant cell organelles. Q24. Which statement correctly distinguishes plant cells from animal cells? A. Plant cells lack mitochondria. B. Animal cells possess chloroplasts. C. Plant cells contain plasmodesmata that facilitate intercellular communication. D. Animal cells possess a cellulose cell wall. Answer, C. Explanation. Plasmodesmata are cytoplasmic channels connecting adjacent plant cells, enabling transport of molecules and communication. They are absent in animal cells. Difficulty. Topic: Plant cell. Q25. A fluorescent dye specifically accumulates in acidic intracellular compartments. Which organelle is most likely to exhibit the strongest fluorescence? A. Nucleus. B. Lysosome. C. Peroxisome. D. Mitochondrion. Answer, B. Explanation. Lysosomes maintain an acidic lumen, pH 4.5, 5.0, making them preferential sites for accumulation of acidotropic fluorescent dyes such as lysotracker. Difficulty. Topic: Lysosomes. Q26. Which of the following is not a function of the Golgi apparatus? A. Glycosylation of proteins. B. Sorting of secretory proteins. C. ATP production. D. Formation of secretory vesicles. Answer, C. Explanation. ATP production occurs in mitochondria during oxidative phosphorylation. The Golgi apparatus modifies, sorts, and packages proteins and lipids. Difficulty. Topic: Golgi apparatus. Q27. The signal hypothesis explaining protein targeting to the rough endoplasmic reticulum was proposed by A. Francis Crick. B. Günter Blobel. C. James Watson. D. Lynn Margulis. Answer, B. Explanation. Günter Blobel proposed the signal hypothesis, demonstrating that signal peptides direct proteins to the endoplasmic reticulum. He received the Nobel Prize in Physiology or Medicine in 1999. Difficulty. Topic: Protein targeting. Q28. Which motor protein transports vesicles toward the plus end of microtubules? A. Dynein. B. Kinesin. C. Myosin. D. Actin. Answer, B. Explanation. Kinesin generally moves cargo toward the plus end of microtubules, anterograde transport, whereas dynein moves toward the minus end, retrograde transport. Difficulty. Topic: Cytoskeleton. Q29. Which statement about the plant cell wall is correct? A. It is composed mainly of chitin. B. Primary cell walls are rich in cellulose, hemicellulose, and pectin. C. Secondary cell walls are formed before cell division. D. The cell wall is impermeable to water. Answer, B. Explanation. Primary cell walls are flexible and consist mainly of cellulose microfibrils embedded in a matrix of hemicellulose and pectin. Chitin is characteristic of fungal cell walls. Difficulty. Topic: Cell wall. Q30. During cell fractionation, which organelle is most likely to sediment first during low-speed centrifugation?
A. Ribosomes. B. Endoplasmic reticulum fragments. C. Nuclei. D. Microsomes.
Answer, C. Explanation. Nuclei are the largest and densest organelles and therefore sediment at relatively low centrifugal forces. Smaller organelles require progressively higher speeds. Difficulty. Topic: Cell fractionation. Q31. Which of the following organelles is directly involved in the synthesis of membrane phospholipids? A. Rough endoplasmic reticulum. B. Smooth endoplasmic reticulum. C. Golgi apparatus. D. Mitochondrion. Answer, B. Explanation. The smooth endoplasmic reticulum, CSIR, is the principal site for the synthesis of phospholipids, cholesterol, in animals, and other membrane lipids. These lipids are later distributed to other cellular membranes. Difficulty. Topic: Smooth endoplasmic reticulum. Q32. Which cytoskeletal component is primarily responsible for cytoplasmic streaming in plant cells? A. Intermediate filaments. B. Microtubules. C. Actin microfilaments. D. Centrioles. Answer, C. Explanation. Cytoplasmic streaming, cyclosis, in plant cells is driven by actin microfilaments interacting with myosin motor proteins, facilitating the movement of organelles and cytoplasm. Difficulty. Topic: Cytoskeleton. Q33. Which of the following correctly describes the function of ubiquitin? A. It acts as a membrane receptor. B. It tags proteins for degradation by the proteasome. C. It initiates DNA replication. D. It catalyzes ATP synthesis. Answer, B. Explanation. Ubiquitin is a small regulatory protein that marks damaged, misfolded, or short-lived proteins for degradation by the 20-SIXS proteasome through the ubiquitin proteasome pathway. Difficulty. Topic: Protein degradation. Q34. Which staining technique is commonly used to observe mitochondria in living cells? A. Safranine. B. Janus green B. C. Acetocarmine. D. Crystal violet. Answer, B. Explanation. Janus green B is a vital stain that selectively stains mitochondria in living cells because it is oxidized in actively resp-iring mitochondria. Difficulty. Topic: Cell organelles. Q35. The fluidity of biological membranes is increased by A. An increase in saturated fatty acids. B. A decrease in temperature. C. An increase in unsaturated fatty acids. D. Extensive cellulose deposition. Answer, C. Explanation. Unsaturated fatty acids contain double bonds that introduce bends in hydrocarbon chains, preventing tight packing and thereby increasing membrane fluidity. Difficulty. Topic: Plasma membrane. Q36. Which cellular organelle contains hydrolytic enzymes but lacks its own DNA and ribosomes? A. Chloroplast. B. Mitochondrion. C. Lysosome. D. Nucleus. Answer, C. Explanation. Lysosomes are membrane-bound vesicles containing hydrolytic enzymes for intracellular digestion. Unlike mitochondria and chloroplasts, they do not possess their own DNA or ribosomes. Difficulty. Topic: Lysosomes. Q37. A mutation in the gene encoding dynein would most directly affect A. Protein synthesis on ribosomes. B. Retrograde transport along microtubules. C. DNA replication. D. Lipid synthesis. Answer, B. Explanation. Dynein is a motor protein that transports vesicles and organelles toward the minus end of microtubules. It is also essential for the movement of cilia and flagella. Difficulty. Topic: Molecular motors. Q38. Which of the following statements regarding plasmodesmata is incorrect? A. They connect adjacent plant cells. B. They allow symplastic transport. C. They are lined by the plasma membrane. D. They are absent from young plant tissues. Answer, D. Explanation. Plasmodesmata are abundant in young, actively growing plant tissues where cell-to-cell communication is essential. They are not absent from young tissues. Difficulty. Topic: Plant cell communication. Q39. In differential centrifugation, which organelle is typically isolated after mitochondria but before ribosomes? A. Nucleus. B. Golgi membranes and lysosomes. C. Chromosomes. D. Cell wall. Answer, B. Explanation. During differential centrifugation, low-speed rightward arrow nuclei, medium-speed rightward arrow mitochondria, higher-speed rightward arrow lysosomes, peroxisomes, Golgi membranes, ultracentrifugation rightward arrow microsomes and ribosomes. Difficulty. Topic: Cell fractionation. Q40. A plant cell is transferred from distilled water to a concentrated sucrose solution. Which sequence of events is most likely? A. Endosmosis rightward arrow increased turgor rightward arrow cell swelling. B. Exosmosis rightward arrow plasmolysis rightward arrow loss of turgor. C. Endosmosis rightward arrow cytolysis. D. No net movement of water. Answer, B. Explanation. A concentrated sucrose solution is hypertonic to the cell sap. Water moves out of the cell by osmosis, exosmosis, causing the protoplast to shrink away from the cell wall, a process known as plasmolysis. Difficulty. Topic: Osmosis and water relations. Q41. Assertion, reason. Assertion: A. Cholesterol increases membrane fluidity at low temperatures. Reason: R. Cholesterol prevents the close packing of phospholipid fatty acid tails during low temperatures. A. Both A and R are true, and R is the correct explanation of A. B. Both A and R are true, but R is not the correct explanation of A. C. A is true, but R is false. D. A is false, but R is true. Answer, A. Explanation. At low temperatures, cholesterol prevents phospholipids from packing tightly, thereby maintaining membrane fluidity. At high temperatures, it has the opposite effect by restricting excessive movement. Difficulty. Topic: Plasma membrane. Q42. Which of the following combinations correctly matches the organelle with its principal function? Organelle. Function: A. Peroxisome. ATP synthesis. B. Lysosome. Intracellular digestion. C. Golgi apparatus. DNA replication. D. Ribosome. Lipid synthesis. AA only. BB only. CC only. DD only. Answer, B. Explanation. Peroxisomes perform oxidation reactions and detoxification. Lysosomes digest intracellular material. DNA replication occurs in the nucleus. Lipid synthesis primarily occurs in the smooth endoplasmic reticulum. Difficulty. Topic: Cell organelles. Q43. A researcher treats cells with cyclohexamide, a drug that inhibits cytoplasmic ribosomes in eukaryotes. Which cellular process is directly affected? A. DNA replication. B. Protein synthesis. C. Lipid metabolism. D. Cell wall synthesis. Answer, B. Explanation. Cyclohexamide blocks translation by inhibiting the translocation step on ADS ribosomes, thereby preventing protein synthesis in eukaryotic cells. Difficulty. Topic: Translation. Q44. Which of the following structures is common to both prokaryotic and eukaryotic cells? A. Membrane-bound nucleus. B. Golgi apparatus.
C. Plasma membrane. D. Mitochondria. Answer, C.
Explanation. Both prokaryotes and eukaryotes possess a plasma membrane, cytoplasm, ribosomes, and genetic material. Membrane-bound organelles are absent in prokaryotes. Difficulty. Topic: Cell organization. Q45. The major function of SNARE proteins is to A. Catalyze ATP synthesis. B. Facilitate vesicle docking and membrane fusion.
C. Stabilize DNA. D. Assemble ribosomes.
Answer, B. Explanation. SNARE proteins ensure the specificity of vesicle targeting and mediate fusion between transport vesicles and target membranes, such as the Golgi apparatus or plasma membrane. Difficulty. Topic: Vesicular transport. Q46. Which technique would be most suitable for observing the three-dimensional surface architecture of pollen grains? A. Transmission electron microscopy, TEM. B. Phase contrast microscopy. C. Scanning electron microscopy, SEM. D. Fluorescence microscopy. Answer, C. Explanation. SEM provides detailed three-dimensional images of specimen surfaces, making it ideal for studying pollen wall ornamentation, trichomes, and epidermal features. Difficulty. Topic: Microscopy. Q47. Assertion, reason. Assertion: A. Mitochondria are regarded as semi-autonomous organelles. Reason: are they possess their own DNA and ribosomes but still depend on nuclear genes for many proteins. A. Both A and R are true, and R correctly explains A. B. Both A and R are true, but R is not the correct explanation. C. A is true, but R is false. D. A is false, but R is true. Answer, A. Explanation. Mitochondria synthesize only a small fraction of their proteins. Most mitochondrial proteins are encoded by nuclear genes, synthesized in the cytosol, and imported into the organelle. Difficulty. Topic: Mitochondria. Q48. During electron microscopy, osmium tetroxide, OSO4, is primarily used because it A. Stains nucleic acids selectively. B. Fixes and stains membrane lipids, enhancing contrast. C. Digests proteins. D. Fluorescently labels mitochondria. Answer: B. Explanation. Osmium tetroxide reacts with unsaturated lipids, stabilizing membranes and providing electron density, which enhances contrast in electron micrographs. Difficulty. Topic: Electron microscopy. Q49. A mutation disrupts the function of aquaporins in root cells. Which process would be most directly affected? A. DNA replication. B. Rapid water transport across membranes. C. Protein glycosylation. D. ATP production. Answer: B. Explanation. Aquaporins are membrane channel proteins that facilitate rapid and selective movement of water across biological membranes. Their dysfunction reduces water permeability. Difficulty. Topic: Membrane transport. Q50. Which statement best explains why plant cells do not burst when placed in distilled water? A. The plasma membrane is impermeable to water. B. The rigid cellulose cell wall resists the internal turgor pressure. C. Plant vacuoles prevent osmosis. D. Water cannot enter plant cells. Answer: B. Explanation. Water enters plant cells by osmosis, increasing turgor pressure. The rigid cellulose cell wall counteracts this pressure, preventing cell lysis and maintaining structural integrity. Difficulty. Topic: Plant cell wall and osmosis.